I'm here to talk with you for the next few tens of minutes about the brain. We've heard a lot of references to the brain today and to computation in the brain and how it relates to computation going on in machines. And that's exactly what I'm going to dive into here. My students always tell me that anytime I give a talk or a lecture, I should start with a big picture, okay? So the big picture is you are here, all right? That's the biggest picture that I know of. At a fairly insignificant solar system at the edge of a fairly insignificant galaxy, but we are here. And from a neuroscientist point of view, the most amazing thing about this universe that we live in is that we can perceive it all and that we can act on it. And everything that we perceive and understand, any bit of sense that we're able to make about our universe happens with this 3 pounds of goo, cereal, porridge up between your ears, this magnificent organ, the brain. And of course, the brain orchestrates any action that you take on your world, including making autonomous cars or reasoning about ethical dilemmas that are coming in the future or anything else. And that is just an astounding fact. To me, it's one of the 2 most amazing facts about our universe. And many of us neuroscientists who do this kind of work for a living were really startled in 2013 at the State of the Union address when President Obama made reference to neuroscience in the State of the Union address, and shortly thereafter, in the first week in April, announced a United States Brain Initiative to actually plumb the mysteries of the brain. And I was actually invited to be co-chair of the planning committee that planned this effort for the United States. That effort was adopted by the White House Office of Science and Technology Policy and also by the director of the NIH, and it's currently being implemented. Now, why would the president and the office OSTP and the White House decide that the Brain Initiative deserved, this kind of prominent status? And I think it was a really smart move on their part, and I would say that whether it was a Republican or a Democratic administration making this announcement, this is not a political statement, but I think it's a smart move because there's a great deal at stake in neuroscience over the next few decades. I think, first of all, it's the grand scientific challenge of the 21st century. As genetics was in the second half of the 20th century, cracking the genetic code, and physics was in the first half of the 20th century, understanding the brain and how it works and how it makes us who we are is maybe the grandest scientific challenge of the 21st century. This one, of course, is on many of our minds once you hit your 65th birthday, the way I am going to next month. Treating neurological and psychiatric disorders, especially those of old age like Alzheimer's, becomes increasingly important to all of us. But I think that neuroscience will give us deep insights into who we are. I think that any of us who live to be, roughly 40 or so have cause to think, why in the world do I behave the way I do? Why is it that I hurt people I love and I am more polite and civil to people I really don't like sometimes than to people I love in my own family? Why? Where does that come from? Where does emotion come from? Where does creativity come from? What are our brains doing when we render moral judgments? But I think neuroscience will also be a big part of the conversation and an actor in who we will become as a species and as people in the future. And as we've heard a lot about already this morning, how we'll interact with technologies and how brain-machine interfaces, for example, have the ability or potential to extend our cognition and our sensing of the world. So it turns out we're not the first ones to want to understand the brain or to pop the lid on the skull and see what actually lies under it. This has been done for 10,000 years. This patient, and I say that in quotes, actually survived this surgery, and I also say that in quotes, as you can tell by bone growth. You can examine this and you can look at bone growth, and we know that this patient survived and lived for several years at least after this particular procedure. And why the Egyptians and the Peruvians were doing these trepanations, we don't really know. We don't really know whether they were really onto something about pressure inside the cranium and releasing it after injury being life-saving in some circumstances, or whether this was part of some ritual. But today, when we pop the top and look under the hood, as it were, very carefully, this is what we see, okay? So this is the brain, and it has the consistency, as I suggested, of tapioca pudding or toothpaste when it's actually in the head. It's densely vascularized, as you can see, and neurosurgeons across the world look at the brain like this every day and doing surgeries and many times in awake people for placement of deep brain stimulation electrodes, which I'll talk about in a few minutes. Our biggest problem, our central problem as neuroscientists, is this problem of reverse engineering the brain. So those of you from engineering backgrounds know perfectly well what I'm talking about. Someone gives you a functioning instrument and says, we don't know what the heck this thing is. Maybe we captured it during some military operation or something. It's your job, Mr. Engineer, Ms. Engineer, to figure out what this is and how it works. And we have a particular challenge because we're looking at a system that's made up of over 100 billion nerve cells. Each of those nerve cells makes 1,000 connections to other nerve cells. So we've got over 100 trillion synapses in the brain. And as a neuroscientist, I look at that and somebody says, your job is to reverse engineer this. And my first question is, where are the knobs on this thing, okay? What do I control? So if someone asked me to reverse engineer this, I'd have a hunch where to start, right? Because this thing here has plenty of knobs on it. We could turn these knobs, we could start pushing buttons, we could examine the behavior of the thing, but what knobs do we have to push on the brain? It's actually a problem. Now, when you pop the top on a piece of consumer electronics, what you see inside is circuits. Okay, and that's a first clue for us because we know that the brain is made of circuits. Those 100 trillion connections are not random. They're not spaghetti. There are well-defined circuits inside the brain, and understanding how those circuits process information and how they interact with each other it's quantitatively, but perhaps not qualitatively dissimilar from a simpler project like this. So over my professional lifetime, which is now, I started studying the brain about 40 years ago when I was in graduate school, there have been 4 critical barriers that have held back our progress in understanding information processing in the brain and how it relates to intelligence and behavior. And the first one, there's going to be a quiz on this, by the way, at the end. So I want you to remember these 4 things. The first thing is simply to map the circuits. Okay? Every engineer has a nice circuit diagram, usually of an instrument that they're working with. We don't yet have a circuit diagram for the brain. It's incredibly complex, and this has been a real holdup. Our techniques for mapping circuits have been really rather primitive, as I'll again tell you more about in a few minutes. The second thing is to measure the signal dynamics. So if we need to get from here to Berkeley, some of us at the end of the day today, you need a map, right? You need a static map. You need to know what roads connect you to Berkeley, therefore what routes you would potentially take. But that's not enough. You really want to know, you want your Google Maps or you want your Waze or whatever your favorite app is to tell you how bad the traffic is on different roads. You want to know, in essence, what the dynamics are within that system. So it's not enough just to have a map. We actually have to be able to study the signal dynamics within the system. The third thing is that we haven't been able to manipulate the signals. So if you are the traffic engineer responsible for traffic in the Bay Area, you really want to know, if I shut down this particular highway at this particular point in time, how is it going to affect the entire system, right? How is it going to affect the signals or the traffic flowing everywhere else? And similarly, if I spend $10 million to build a new stretch of highway, what benefits is that going to get me for the system? So you actually want to know what's going to happen to the whole system when you manipulate signals in certain particular regions of the system. And that is a critical thing for reverse engineering. And then, there's finally, get a theory for crying out loud, right? So we can acquire data in great gobs now, huge, gigabytes of data in single experiments, and it won't be too long before they're up to terabytes. But without a theory, we simply won't understand what's going on. We'll get lots and lots of data, and the big failure mode, potential failure mode, is at the end of 10 years, with all that new data, we'll just be 10 years older. Okay? And we won't really understand what those data mean. So we have to work with theorists, with statisticians, with computer scientists, engineers, to develop adequate theories for neural computation. So these have been 4 critical barriers, but the great news is that these barriers now are falling on all fronts. And this is a critical reason why the Obama administration decided to launch this BRAIN initiative, because they sense, and I think there's a general sense within the field, that revolutionary things are taking place right now because we're making rapid progress on all 4 of these fronts. The new technologies that have been developed, several of them here at Stanford, as you'll see in a moment, are making possible experiments and manipulations and the acquisition of data sets that we simply have only been able to dream or fantasize about in the 40 years of my career. And what I'm going to do in the next 10 minutes, 12 minutes, is take you on a Cook's tour of these forefronts. I'm going to give you insight into the new technologies that are making the circuit mapping so much more effective. I'm going to tell you how we're measuring signal dynamics in new and revolutionary ways. And I'm going to tell you about new techniques for manipulating very specific signals in the brain and testing their effects on behavior. And I'll just have a few words to say about where I think the frontiers are in theory. So those are the four things. That's your roadmap here. We've heard that word before, but I'll ask you about them as we go along. So let's talk about circuit mapping first. So this is a mouse brain. Many experiments in neurobiology are done in mice. Some are done in primates. Some are done in lower species like worms and flies. But this is the same mouse brain sitting right over here. And this mouse brain has simply undergone an electrochemical clarification process that basically leaches all of the lipids out of the brain. And the lipids are what makes the brain opaque, what does light scattering. And once you get rid of all the lipids, you can actually see right through this brain. Now, before the lipids were extracted, a hydrogel was created that actually cross-linked all the proteins and nucleic acids so that the cell structures and the locations of molecules were actually held in place while the lipids came out. Now, the big reason for desiring a transparent brain like this is that if we wanted to trace the circuits in the old days, when I was in graduate school in the first 20 years of my career, what we did was we took an instrument called a microtome and we cut the brain into very thin slices, like a loaf of bread. And we would very laboriously mount each slice on a slide. Then we would stain it with particular stains and look at them under the microscope, one slide at a time, and try to build a 3D picture of the circuit from examining the slices one at a time. Now, with a clarified brain like this, what you can actually do is osmose in antibodies or other chemicals that actually have fluorescent labels and label particular pathways that are designed by neurotransmitters like dopamine or like serotonin. And you can follow and map out the circuits for any particular neurotransmitter optically, simply by putting this clarified brain on a microscope stage and then focusing down optically in actually tracing the fibers to see where they go. And it renders obsolete this very laborious method and increases the power of circuit mapping by at least an order of magnitude. And there are improvements that are on the side in our headlights now that will increase that probably by another order of magnitude. Now, this invention, or this particular way of doing it, called clarity, was invented by Deisseroth in his lab here at Stanford, published in Nature 3.5 years ago now. And in that issue of Nature, there was a video that appeared, and this video here is worth 1,000 of my words. So let's just watch it, and it will give you a little notion of how powerful the clarity technique is.
We're heading into the center of a mouse's brain, into the hippocampus where memories are formed. Looking up, you can see neurons projecting to the surface of the brain. This is the work of Deisseroth and his team at Stanford University. By making the entire brain transparent, they were able to image it using a light microscope. They call the new technique clarity. Existing techniques for studying the brain's wiring are often limited to looking at very small volumes of brain, or they don't allow you to label genes or chemicals of interest. The advantage of clarity is that you can label lots of molecules in whole brains.
So we'll stop that there, but I think that gives you an idea. I mean, these are not animations or artist conceptions. These are real data acquired in 3 dimensions in the brain with a real ability to track those axons and dendrites and see what connections are being made. And it's a remarkable advance in our ability just to watch and recreate and map these static connections and circuits within the brain. So that sort of mapping the circuits was the first big barrier, right? What was the second one? This is your quiz. Yeah, measuring the dynamics, measuring the signals. Okay, now this is an incredibly cool paper, again, that was published in 2013. It's from Misha Ahrens and Phil Campbell and their colleagues at Genili Farm Research Campus at Howard Hughes Medical Institute. And what you're looking at here is the head of a larval zebrafish. So this isn't even a full-grown zebrafish. This is a tiny little larva that's about 1 millimeter or 2 long. And you're looking down at the head, and you're seeing the brain here. And the body of the little fish goes off in this direction. Now you would say, why does anyone want to study the brain of a larval zebrafish? And the answer is that it has a magical property, and the magical property is that the skin of the larval zebrafish is transparent. So in the living animal, you can actually look through and see the brain and image the brain with a microscope, which is exactly what you're looking at here. This is the outline of the animal's head that you're looking at. This animal is immobilized in agar. It's like jello, so it's immobilized, but it's living, it's breathing, respiring, and most importantly, all of these nerve cells are alive and well and functioning. This animal has been transgenically modified so that every nerve cell contains a gene and a protein product that fluoresces when that cell is electrically active. So all of these little orange-yellow spots you see are active neurons, and the sort of pale gray material are neurons that are not active. And you can actually sit here and make movies of an actual signaling nervous system, and I'll play this for you. And for people of my generation, for whom we studied every cell one at a time very carefully with microelectrodes and built up a post hoc picture of this kind of population activity after the fact, a year or two of experiments, this is nothing short of astounding. And there are these big bursts occasionally like that happened. I'll play that for you again. And I don't know what in the world that big burst was. Was it a zebrafish thought? Was it a door slamming? Was it a seizure? We don't know, but I'll show it again. But there are regular features here. You can see there's a couple of populations of cells here across the midline that are firing in antiphase with each other. You can also tell that there's occasional signals coming down here to the spinal cord. There is the big thought, the big idea of the zebrafish. But the astounding thing is that you can acquire, these guys can record from about 80,000 of the 100,000 neurons in the zebrafish brain in one experiment. And that was inconceivable to even imagine being able to do that 5 years ago. It's just astounding. And what we now have to do is to figure out how to do careful analysis of this to figure out which neurons are working together, co-varying, which ones are working in opposition to each other, which circuits seem totally to have nothing to do with each other in mathematical sense. Their activity would be orthogonal. So we can get the circuits, and we are making huge progress on getting these kinds of signal dynamics. Now, what was the third tool that we wanted to do the reverse engineering? Yeah, we want to manipulate those signals, right? We don't want to just look at it. We want to reach in there and turn a knob and say, I have an idea about what one particular circuit might be doing. I want to turn that circuit up in its activity, or I want to turn it down, and I want to see what difference it makes, okay? But before you actually go to that kind of manipulation, another thing that you really want to add on top of what you have with this zebrafish right here is activity, is behavior. So this animal's immobilized, and we might develop ideas about how those circuits relate to each other, but unless we actually have the animal executing some kind of behavior, we don't have a way of testing that. So Mark Schnitzer here at Stanford in the Department of Applied Physics and Biological Sciences actually built a little microscope that sits on, it weighs 4 grams. It can be mounted on the head of a mouse that's navigating in some kind of environment like this, hooked up by a fiber optic cable, and has a tiny lens at the end of a fiber optic probe. And you can actually image populations of neurons virtually anywhere in the brain through this little lens and fiber optic that's actually mounted directly on the skull, and you can measure these cells while the animal's solving mazes, while he's searching for food, while he's interacting with other animals, etc. So putting the behavior and the dynamics together is really critical. Now, to actually manipulate the circuit activity, a new technique that was invented again from Carl Deisseroth's lab here at Stanford about a little over 10 years ago now, called Optogenetics, is revolutionizing the neuroscience world. It has turned it on its head over the last 10 years. Optogenetics is a way to control the activity of specified circuits of neurons with light, thus the name optogenetics. Using the magic and trickery of genetic modification, you can actually label, or you can actually impregnate certain cells and circuits within the brain with these proteins that sense lights. They absorb photons, but when they absorb a photon, they open an ion channel in the membrane so that current flows in or out of the cell. And you can actually power the cell, that is, make it become more active by shining one wavelength of light, and you can cause an inhibitory current that actually suppresses the cell or the circuit by shining a different wavelength of light. So these are remarkable new tools made possible through the genetic revolution and through our increasingly sophisticated ability to apply those tools to the nervous system. So this shows one of these plugs. It's a little fiber optic that comes on that is shining light into this animal's brain, and it'll be shining light on the specific groups of cells that the scientists are interested in studying. And I'm going to show you a little video clip of optogenetics in action. So this is going to be optogenetic stimulation of a little nucleus in the amygdala, the medial amygdala, which is a structure that we know to be involved in controlling emotional behavior and aggressive behavior and filial kind of pro-social behavior. This was done in David Anderson's lab at Caltech and just published in the journal Cell 2 years ago. What you're going to see here is a pair of mice and what's called the unfamiliar intruder paradigm. So there's a male mouse, and you'll see him with the fiber optic going attached to his skull. And the male mouse is sitting in his cage, and a female mouse, who he has no prior familiarity with, is introduced into his environment. And male mice, not surprisingly, in the presence of a novel female, are very interested and very curious and generally very friendly. But you'll see what happens when this circuit gets turned on optogenetically in the amygdala. You'll see the mouse's behavior change rather radically. So you'll see the words light on or laser on appear, which is the moment they start shining light in the amygdala, and you'll see the change in the animal's behavior. So this is channel rhodopsin 2 stimulation of medial amygdala. So here's our male mouse right here with his fiber optic. He's very curious about this female. He doesn't know. The laser gets turned on, and suddenly he goes into very aggressive mode, the way he would an unfamiliar male who had been introduced into his cage. So you can see these are very aggressive interactions. And then the laser gets turned off, and he simply goes back to being a very curious male mouse interacting in a very normal way with this female. So this is an example of the power of our ability that is developing now in genetically tractable organisms to actually manipulate circuits and turn them on and off, and in this case have a very dramatic effect on behavior. In other cases, there will be much more nuanced and much more subtle things. So these tools are coming, and as I said, we could only dream about that for most of my career, for the first three decades of my career. We had a set of techniques we could use for manipulation, but none were as genetically precise and as beautifully targeted as these kinds of manipulations. In the theory range, in the theory area, I'll just say, so for the mapping the circuits, I've illustrated clarity for you. In signal dynamics, there are new forms of microscopy and calcium imaging. Both of these genetic manipulation is critical for. And then manipulating the signals, optogenetics has some cousins. So we also have a new generation of pharmacogenetics that are giving us new tools for manipulation. In the theory area, what we need is more than anything else, we need applied physicists, we need engineers, we need statisticians who are good at theoretical and conceptual approaches to nonlinear dynamical systems. The brain is nothing if not a complex dynamical system, and it is very definitely nonlinear, starting right, for those of you who speak this language, following right at the individual synapses and individual neurons. There are profound nonlinearities. And people like me, sort of classically trained biologists and neurophysiologists, I know how to get these signals. I know some good questions to ask about the brain, but I don't know how to think about it at a holistic computational level as a nonlinear dynamical system, but I have collaborations with engineers right now that are making those things possible. And this is a real growth area in the field, but it's behind the other three and definitely needs to catch up. So these are the 4 critical barriers that are falling as we speak, and just tremendous progress has been made in the last decade, which is why many of us sense that we're at an inflection point. And for those of us who are professors, we have to actually step back and say, what is it going to mean in the future to be a neuroscientist? And what do we need to teach our students to enable them to have the largest impact on understanding the brain? And it's causing a lot of re-examination. And one of the things I'm dead sure of is that solving the brain is no longer just a problem in biology or medicine. Truthfully, it never was, but it was often treated that way. It's often in medical curriculum somewhere, right? In medical schools typically. But you can see just from this list right here, this is an all-hands-on-board effort, right? We've got to have the geneticists, the chemists, we've got to have the physicists, the engineers, the computer scientists, as well as the traditional neuroscientists. Now, what's the payoff? What's the promise? If we get this kind of reverse engineering approach going and we get some circuit-based knowledge of the brain and behavior, one of the real payoffs, as I suggested earlier, is circuit-based interventions in disease. So many of you have probably heard about deep brain stimulation. There are about 100,000 patients with pretty advanced Parkinson's disease who have had these electrodes put in their brain, 2 of them, 1 on each side, and targeting an area called the subthalamic nucleus. The subthalamic nucleus is a part of a larger circuit of structures called the basal ganglia that interact with the thalamus and with the cortex. And these basal ganglia, these circuits were mapped the old-fashioned way, the way we did it when I was in graduate school. We earned our data, right? We sliced up the loaf of bread. We didn't have all of this magic stuff going on. But the STN has been the target from our understanding of the basal ganglia and its dysfunction in Parkinson's disease. It's been the target of these kinds of circuit-based interventions. And you can basically think of this as a pacemaker for the brain. So the patient wears a little pacemaking battery under the clavicle, just like for a heart pacemaker. These 2 electrodes are inserted deep into this target, the subthalamic nucleus in the brain, and basically pulses go into this part of the brain 100 per second, 24/7. And the effects are nearly instantaneous when you start the pacemaker going, and when you turn it off, the symptoms of Parkinson's come back almost immediately. And I just want to show you a little video material from this patient here. Her name is Sybil. You can actually find her on YouTube. You can find these videos that I'm going to show, and you can find many others like them on YouTube. But I'm going to show you a before and immediately after and a little bit after a second surgery and show you the power that deep brain stimulation can have.
Even a simple task like eating breakfast was a frustrating battle with her own body.
How can you live your life when you're shaking so much?
It's extremely difficult, sometimes even overwhelming. And I get very emotional. Sometimes I cry. It's just a hard thing to do.
The disease had gotten so bad that sometimes her muscles froze completely, making her face almost expressionless and her legs almost useless. The woman who once was always on the go could barely move, confined to a wheelchair.
So she had her first surgery. This surgery was done at Emory University Medical Center about 12, 13 years ago, so kind of in the early days of deep brain stimulation. They were more cautious than they are actually now, and they put 1 electrode in and started stimulating, and you're going to see them you're going to see the doctors interact with Sybil like it's only 1 or 2 days after the first surgery.
Days after the operation, in a moment that would test Sybil's dreams.
Let's see how you do.
After 2 long years trapped in a wheelchair, would she finally be able to walk?
Right arm is swinging.
So the right arm swings. Yeah.
I haven't swung my right arm for I don't know how long.
So the reason she could swing her right arm and not the left is because she only had 1 electrode in, stimulating one side of the brain. So they did it. This one, they had such a good result from this one that they went in and put the second electrode in, and they catch up with her now 2 or 3 months after the second surgery.
A month after her second surgery, when we caught up with Sybil again.
So Sybil, how do you feel?
Great.
Remember that wheelchair? It was gathering dust in the garage. Doesn't have to.
What can you do now that you couldn't do before?
Oh,
Good heavens, look at that! Look at her! Look at that!
Sybil and Alvin had hoped for a lot from the surgery, but the outcome was better than they could have imagined.
So this is an extraordinary result here, right? But I have to tell you a couple of things. First of all, it's not that effective in everyone. So people are carefully selected for deep brain stimulation. You have to be in good overall health for the surgery and such. Secondly, there's a range of effects. There are many people for whom you get excellent effects like this. There are some people for whom it's not so excellent, but you can tell by the fact that 100,000 have been done now that this is incredibly effective. But the biggest caveat is that this is not a cure for Parkinson's disease. So the fundamental pathology in Parkinson's disease is that a group of cells in the brain called dopaminergic, they have the chemical or neurotransmitter dopamine, those cells start dying. We don't know why they start dying. And the symptoms of Parkinson's progress with the death of these cells. What you do with deep brain stimulation is that you buy somebody 2 or 3 years of their life back. Okay? But the cells keep dying, and ultimately that brain stimulation will lose its efficacy. But right now, it can be a very critical bridge to giving people some years of useful life that they would not have otherwise. And that is the promise of understanding these circuits. If we can actually start understanding how they work and why they work the way they do, we can design these kinds of interventions. So deep brain stimulation now is being explored as treatment for obsessive compulsive disorder, for Tourette syndrome, and a number of others are in clinical trials as well. The ideal, of course, that we'd love to get to, and again, this is going to come when it comes by pushing technology, the ideal that we'd love to get to is to be able to stimulate these circuits with fine spatial and temporal resolution non-invasively, right, from outside the brain. If I were king of the United States, that used to be more funny than it is now, but if I were king of the United States, I would take one of our big national laboratories with so much brain power and technological horsepower, and I would add some biologists to that national laboratory, and I would say, your mission over the next 10 years is to find a way to stimulate and record from the human brain safely and non-invasively. And if we could do that, we could do so much clinically for people and create so much understanding about where our actions come from. I'll give you just one example. Our treatment of last resort right now for truly life-threatening depression is ECT, electroconvulsive shock therapy. And basically, ECT shocks a large region of the entire brain, and it's really effective for some people with chronic depression. I have had 2 friends of mine, 2 personal friends who have had this, and their depression has been really helped. Unfortunately, because it's kind of a shotgun approach, it's not precisely targeted, you get major side effects, mostly on memory, short-term memory and such. So I think that if we could have a non-invasive technique where we could target real specific circuits, we could probably just have people come in for their treatment 3 or 4 times a year and manage that kind of depression with very few side effects. And that's the kind of thing we could put ECT on the trash heap of medical history, and that's where it belongs in the long run. Okay, so just remember, just my parting memory here is that there's a lot at stake in this effort. The grandeur of a remarkable scientific challenge, but being able to treat neurologic and psychiatric disease across the entire lifespan from birth until death, deep insight into who we are, where our behavior comes from, and then influencing this conversation that, as we've already heard this morning, and we'll hear more this afternoon about who are we going to become as our persons, our bodies, our brains interact with new technologies. And this kind of highly interdisciplinary effort, the fact that this is required is why President Hennessy, former President Hennessy and former Provost Etchemendy started the Stanford Neurosciences Institute 3.5 years ago. We're now in our 4th year of operation, and it is precisely our task to provoke collaboration, provoke conversation, provoke dialogue among all of the schools and departments at Stanford to solve and address these deep mysteries of the brain and the human person. Thank you very much.